Now, continuing on this journey, I'd like to bring up Acacia Mayfield of Asymptote building a universal gene therapy for KCNQ2 DEE to round out our brilliant people telling us about what the future might be for KCNQ2. Thanks a lot for joining us.
Thank you guys all. I'm especially thankful to the organizers for including our small young group. Just to introduce our company, we are Asymptote Genetic Medicines. We are a spin-out, very small organization from Caltech, and you've heard from a lot of people who are part of very established teams from large organizations that are very advanced. We actually haven't even come up on a year yet of our own existence, but we really think that the most important thing is to come and learn directly from the patients. And so the opportunity to come and meet you all and to learn from the other people who know much more about all of this in the field has been a great honor and a very humbling experience. So I'm looking forward to getting to know more of you and to get some input on what you think that we can do to help out in this field. So my name is Acacia. I am from Caltech. I'm originally from my PhD work, and some of the science that I developed and the work in my PhD, I'll present just a little bit here, but really what I want to contextualize through this talk is where this modality is maybe a little bit different from some of the other things that you've heard about already. So what we want to do, which is admittedly a very difficult problem, is we want to take this, which is, the difference between how all of your children are experiencing the disease, and develop something that we hope would work regardless of what the mutation is. So I'm going to tell you a little bit about how we try to do that. So as some people have already mentioned, there are different ways to address disease. Some interventions are aimed at looking at how the neurons in the brain are responding within their own collective and trying to interfere, in the case of epilepsy, with how the cells are firing together inappropriately. Other therapies are aimed at saying, well, why are those cells firing incorrectly? If we can't control things at that level for all patients, maybe we can go a level below and try to modify the activity of a protein specifically. And you can also go one step further if you're talking about a genetic disease. So what genetic diseases are is you have a known sort of root cause of a problem that's causing this constellation of symptoms. And what we aim to do in genetic medicines is to intervene at that point. Intervening at that point, theoretically and ideally, we hope would impact the whole spectrum of what the patients are experiencing. So how does genetic disease actually turn into what a child is experiencing in their life? When we think about gene expression, Brooke really had a beautiful illustration of how genes turn into the active thing that is biologically making our experiences happen. So normally, you have a healthy gene, which is creating a protein that is functional and is working together like a symphony or like ingredients in a recipe in order to bring some sort of desired function or desired product. And what happens when you have a genetic mutation is that you can either have mutations that stop that gene from being produced, so you're going to decrease the level, and that throws off the recipe, right? If you forget to add the sugar, you forget to add the salt, you get something very different. You can also have mutations that cause a gene to be produced, but it doesn't function in the way that it's supposed to. So you're throwing something into a system that maybe is going to throw off the rest of the symphony, and that can cause, on the level of a cell or on the level of an individual, the reasons why you have these manifestations that can be detrimental. So what gene therapy aims to do, and there are many different kinds, but essentially aims to decouple the context of having a disease gene with a mutation and saying, well, we have this disease. You can't choose the genes that you get, and you get what you get. And what gene therapy tries to do is to say, okay, you have this context, but maybe we can intervene and provide either an extra copy of, like a clean copy of a gene or some other method so that we can restore at the cellular level this harmonious team of proteins that work together. So genetic medicines are made of multiple parts, and ultimately, sorry, this is a little bit backwards, it's sort of the same if you conceptualize it as taking a pill, and in some ways very different. So if you're taking a traditional medicine, you have a delivery mechanism, which could be an encapsulated pill, it could be a liquid suspension, and then you have the actual drug that's going to be doing the thing in the cells that is trying to fix a function. And then you package that together, and that is the medicine. So in gene therapy and genetic medicine, we have a similar concept. We have a package, which in our case, and largely in the field, is a capsid or a delivery mechanism that's going to help our genetic medicine, our cargo, which is the thing that's performing the function, get into the cells where it needs to go. Together, that gets delivered to the patient, and through different mechanisms, depending on the technology that you use, you can either basically stick a sticky note, post it onto the grandma's recipe and say, okay, here is an extra copy, an extra side dish, that we can then have functioning along the rest of what the cell is doing. There are other ways to do this through editing. As Kara mentioned earlier, you can go in and you can make notes or edit grandma's recipe directly. There are different technologies and different considerations that go into each of these, but what we are working on is largely in the first category. So what does this mean for KCNQ2 DEE? So what brought us together in this group is that we noticed that KCNQ2 is among a spectrum of disorders that has this very interesting issue. There is a spectrum of mechanisms. So even though each of these children has a mutation in the same gene, the disease of each of your children may be caused in fundamentally different ways from each other. And we think that this is an interesting problem, not only for drug development, but also for, what is the level where one intervention for a diagnosis is going to be able to address each of those mechanisms. So as you all know probably better than I do, you can have a healthy channel, which is moving potassium out of the cell. You can have a channel where you have a mutation that makes it hard for potassium to move appropriately. Or you can have other types of mutations where maybe you have a hyperactive mutant where potassium is moving in some cell types inappropriately, and that may be causing the whole system to be more prone to overactivity. And so when you have issues that, you have a disease that is a mutation in the same gene but is causing basically opposite problems, how do you address that with a single therapy? So what we're aiming to do, admittedly, is a bit ambitious, but we want to be able to leverage technology that we have been able to show works in other contexts in order to say, what if we can just clear the board? What if we can say, okay, maybe you have an overactive mutation, maybe you have an underactive mutation. What if we can start fresh in all the cells that receive a clean copy and reduce the expression of the mutants, replacing it with a known and constant copy of a healthy KCNQ2. The goal here is to mitigate mutants regardless of what the mutation is, leaving no patient behind, and that it would be a one-time therapy and that it would restore balance to the brain, starting at the very root cause. Hopefully, this would help to alleviate not only seizures, but other endpoints that we don't necessarily understand the biology of at this point. So this is very challenging. There are many layers to how we build a therapy like this. One of them is with our core technology, which is circuit regulation. So our company is founded based on this technology that uses microRNAs within our therapy so that while we deliver the gene of interest, which in this case would be KCNQ2, we want to make sure that because we know that overexpression and overactivity can be a problem, we want to make sure we're not putting back too much of that into any given cell. We want to put back the right amount. The other thing is we want to make sure that we're delivering to the cells that need to receive the gene. And overall, when we combine those things together, we want to be able to create balance. So our vision for our company is that we're trying to make precision therapies. We want to get the right gene into the right cell at the right level. And this is a hard problem, but we have assembled a group of people that are very gracious in advising us and leading us. Our leadership has experience in industry and moving drugs, genetic drugs forward through the development process and more traditional biologic pharmaceuticals as well. And we were born out of an academic project. So our academic founders include Michael Elowitz, who's a synthetic biologist, and Viviana Gradinaru, who was my advisor and who is a leader in the field of delivery. We also have engaged with many other people who are important for the whole process. As we've understood and has been highlighted at this meeting, we need to be thinking very far ahead if we're going to make this work. So our foundational work is in a disease that is called Rett syndrome. So this is not KCNQ2, it's not even an epilepsy, but what it has in common with KCNQ2 is this disorder, it really matters the dose. Like, you can have too little of it, and that causes a developmental disorder, autism spectrum disorder called Rett syndrome. But if you so much as duplicate that gene naturally in a child, you get a corollary disorder, MACP2 duplication syndrome. So we know clinically that it's very important that if you're going to give this gene back, that you have to give it at a very controlled level. And what we were able to do is to tune a set of our core technology circuits in order to see what is that level and test it in mice. And what you're seeing here, there are a couple of takeaways. The black line is the performance of a normal mouse across multiple domains of performance. The red line is an untreated disease model mouse. So that's the sort of phenotype that you would expect. What we were able to see are two major things. Number one, we're able to recapitulate that if you overexpress, if you deliver way too much of this MECP2 into a disease mouse, you basically get the same MECP2 duplication phenotype. You get disease even though you gave back the gene because you gave the wrong amount. So that is the gray line. And then we saw that if you can quantitatively control and rein in the level of replacement, then you can get much, much, much closer to what a normal mouse behavior should look like. So this is just to illustrate that we are able to achieve quantitative control at a molecular level in a way that we think should be applied and moved into other fields of disease. And we're not the only people who have thought of this, so it may be the first time that you're hearing about this, but there are actually two companies in the Rett syndrome field who have put this to the test and have seen clinical benefit in patients. And these trials are ongoing and they're emerging. But we're thinking about what impact does this make to the patient? How does controlling this expression actually matter when you actually go in? And what we're seeing from both of these trials, which are not using exactly the same approach, but are using a similar approach to what we're doing, The Neurogene has treated patients at a variety of age ranges, and they're seeing patients go from a baseline where they don't expect a lot of improvement, no use of the hands to being able to self-feed, poor balance to being able to sit on their own, poor coordination to being able to participate in complex activities. Taysha is seeing very similar multi-domain improvements. And so these sorts of things are basically why we're here. We want to know from you, what does it take? What do you need to see? And how can we be preparing to measure that later on? So that's for MECP2. KCNQ2 is slightly different. There's 2 challenges, whereas for Rett syndrome, there's only one. Because our approach, we want to make it universal to all the mutations. Not only do we have to control the replacement or the supplementation at the level that we think is necessary for function, we also have to get rid of that endogenous expression. So we're trying to do something extra here in order to make it universal to the whole patient population. So what have we done so far? Like I said, we're not even a year-old company, and we're working very hard. So far what we've achieved is we can achieve controlled expression in a mouse model of KCNQ2. We're using the T274M model that was mentioned in a couple of other presentations. And we're undergoing several rounds of engineering and evaluation of the endogenous knockdown system. That's ongoing. We've done some preliminary testing in mice towards the seizure endpoint. We are very cognizant of that is not the only manifestation that matters for KCNQ2 DEE. So we've tried to add in some other measures, but we want to hear from you also what else is the most important. So we came here to learn. I'm very, very honored to be invited to speak as well, but really the experts in the room are you. What do you need for this therapy to make a difference in your lives? And we'll try to build it. So thank you again for welcoming us into your community. It's an honor to be here and to meet all of you, especially to Brooke Babineau and the rest of the KCNQ2 Cure team for facilitating the connection. To Ingo Helbig, who is an epileptologist at CHOP over here in Philadelphia. He's been very instrumental helping us to understand genetic epilepsies and different sorts of things to consider as we try to move forward. And of course, our academic founders and team. I didn't work alone on my projects. None of this would have been possible without my colleagues. And of course, to the patients and their parents. We're looking forward to having a great relationship with you and the community.